So this is lecture 3 of Principles of Metabolism, and in this lecture we will look at oxidation and reduction, which are two very important processes in metabolism. So you might almost say that metabolism consists almost entirely of oxidation-reduction reactions. There are other kinds for sure, but this is a very important part, and you will see this all the time. So a bit sort of simplified, you can pretty much say that catabolism is the oxidation of carbon. So breaking down things and extracting energy is the oxidation of carbon. And anabolism, which is the biosynthesis of things, is the other way around, that is reduction. So what is oxidation and reduction? So probably the most common oxidative process people have seen is the oxidation of glucose. I think you learned this in high school or even earlier. This is a classic case where carbons are being joined with oxygen to form carbon dioxide, and this releases energy. And so this is a major process used to power cells. But so why does this release energy? Why is this an energetic process? So what's really going on here is that carbons are donating electrons to oxygen. And this is a favorable process. And we will look a bit more closely at why this is favorable and why this happens. When we talk about electrons, of course, we generally don't mean electrons flying around in free space in cells. They are usually carried by something, by NAD, NADP, these common energy carriers. But we'll not think so much about that right now. We'll focus on what oxidation and reduction is and where these processes go. So one important concept, this is kind of physical, but I think it's important, is electronegativity, to understand why this happens. So electronegativity is a property of atoms to attract electrons. And it's simply a fact of physics that some atoms are more strongly attractive for electrons. So electrons prefer those which are called more electronegative. And there's a series of these, and carbon is somewhere in the middle along with sulfur in electronegativity. Oxygen is very electronegative, so it prefers electrons, it tends to attract them. Hydrogen and phosphorus is on the other side. They tend to want to get rid of them. So all processes where electrons flow towards oxygen or nitrogen tend to be favorable. And this is the basis for why oxidation releases energy, really. So this can be quantified, and we will see this is kind of a useful concept, something called the oxidation state. And we usually focus on carbon because this is really the most important element in metabolism in this respect. So the oxidation state of carbon. So if you think about a carbon atom, it binds 4 other atoms, whatever it might be, when it sits in a molecule. If you think about the total number of electrons that it donates, gives away, or receives, the oxidation state quantifies this. So the oxidation state is positive if the carbon atom has donated a bunch of electrons, if it has become oxidated, or it is negative if it has received electrons, then it is reduced. So you can quantify this into a number, the oxidation state. So you can calculate it if you look at a molecule as the number of bonds that the oxygen atom shares with either nitrogen or oxygen. And then you can subtract the number of bonds that it shares with hydrogen or phosphorus. So what do we end up with? So let's look at our functional groups. We looked at these in the introduction lecture. Look at some common functional groups. So if you have groups of this type, methyl groups, straight carbon change, they carry a lot of hydrogens, which means that they will be negative. They have a lot of bonds to hydrogen. The extreme case would be -4. That's just methane, but that doesn't occur so often. So generally the the lowest oxidation state we see is methyl groups at -3. So this is highly reduced. And you can say that this is energy rich in a sense, because here there's a lot of potential to oxidize the carbon and extract a lot of energy. And then as you go down the list, you encounter functional groups that have fewer hydrogens bound, that have more nitrogens and especially more oxygens bound. And as you go down the list, you come down to CO2, which is really the most oxidized state you can reach, because here all the bonds of the carbon atom are shared with oxygen, which is the most electronegative atom. So now you are in the state of +4. So all the 4 bonds of the carbon atom is shared with oxygen. Highly oxidized, and now is energy poor. So this is kind of the series. Let's look at these functional groups a bit more carefully, and let's look at some. So here are some common metabolites exemplifying each of these states and functional groups. So at the most reduced state, as we said, we have methyl groups, CH3 groups at the state -3. These you find, for example, in choline. We have 3 methyl groups here. So this is a metabolite that occurs in phospholipids and so in membranes. So to synthesize this, you need to reduce carbon quite a bit. You also see methyl groups in methionine, and we know that there's a lot of methylation of DNA and proteins. And so to synthesize those groups, you need to have very reduced carbon. So this is highly energetically costly things to synthesize. The next step would be the straight carbon chains. So probably the best example of this is saturated fats. So this is palmitate, for example. It's a good example of this. And this is the reason really that saturated fat is such an energy-dense nutrient and storage form of energy, because there's great potential here to oxidize carbon a lot and extract a lot of energy. So that's helpful to know. The next step would be the double bonds between carbons, structures that look like this. So now you're at state -1. And these we see in unsaturated fats. So arachidonate is one example, and there are many of these. So there's a bit of a middle ground. And then we get to state 0. One example of this is the amino acids. So that would be the amine groups, looking like this. I put a star here on this state, so 0 star. So it's pretty much the same as alcohol groups, which would be the next rung on the ladder. But nitrogen is a bit less electronegative than oxygen. So, you know, it's a little higher up, you might say. This depends on which text you read. Some authors consider these to be identical. But generally, I think it's a bit higher up. But so here you find amines. So you find things like glutamate, which is an amino acid, and of course all the other amino acids have the same groups. There are also other biological amines like histamine, for example, that contain these type of groups. The next step on the ladder are sugars, pretty much. So hydroxyl groups, alcohol groups, are present in all sugars. So glucose is the archetypical sugar, of course. And it's interesting to note that this is kind of in the middle of the series. So sugars are not the most energy-rich substrates that you can find. They're kind of on par with amino acids. But they are still better substrates for oxidation for other reasons, because oxidizing amino acids gives you ammonia, which is a bit of a problem practically for the cell. But in terms of energy, these are kind of on the same step of the ladder. And now we're starting to move down towards more oxidized things. So aldehydes is the next group. These are things that look like this. It's an oxygen and a hydrogen bound to carbon atoms. These tend to be reactive, and they tend to be intermediates in pathway. So glyceraldehyde is one example. This is actually a sugar. And actually glucose has an aldehyde group as well, but only 1 out of 6. But these are somewhere on the middle of the ladder. As you move down, you get to ketones. So an example of a ketone is the ketone bodies, acetoacetate, which has this ketone group. You also find these as intermediates in fatty acid synthesis and in fatty acid oxidations. You see these type of structures. So this is somewhere in the middle of energy metabolism, you would say. And then we get to the carboxylic acids. And these type of organic acids are very common in central metabolism and in the TCA cycle. So citrate is an excellent example. This has a lot of these carboxylic acid groups. There's 3 of them, in fact. So these are now at state +3. So this is pretty oxidized. And you can almost recognize that, this is almost carbon dioxide. If you change one bond, this will split off. So now we are down towards the bottom. And then the final state is carbon dioxide, and now you're at +4. And this is as far as you get. So these oxidation states can be helpful to predict which way is the reaction really going to go. If you're studying an enzyme of interest, or you're looking at some reaction in a pathway, in which direction will the reaction go spontaneously? So here's an example. If you look at the reaction between pyruvate and lactate, this is an oxidation reduction reaction, and what's really happening here is centered around this carbon, which forms a ketone group in pyruvate, but which forms a hydroxyl group in lactate. So there's a change in oxidation state. This carbon is +2 in pyruvate, but it's 0 in lactate. So as we said, carbons like to get oxidized. They like to donate their electrons to hydrogen or nitrogen. And therefore, this reaction you would predict to go in this direction towards the left. It is unfavorable in the direction left to right, as written here. So you would say that if you want to make lactate from pyruvate, we predict just from the oxidation states that you would have to put in extra energy in order to make this happen. And that is indeed what happens in lactate dehydrogenase. You need to use the energy of an NADH molecule in order to make this go left to right. So that makes sense. Another example is the oxidation of alpha-ketoglutarate. This is in the TCA cycle, one of the reactions there. So this is a favorable reaction. We can see that because there are increases in the oxidation numbers of carbon. There's 2 of them, in fact. There is this group, a carboxyl group in alpha-ketoglutarate, which splits off as carbon dioxide, so it goes from +3 to +4. That is favorable. But there's also another change, because since we have cleaved off one group, one carbon, this carbon now, which is a keto group in alpha-ketoglutarate, becomes a carboxyl group in succinate, which is this part. So this is favorable and it proceeds to the right. And below here we see another example of the peptide bond. Peptide bond formation is kind of a borderline case when you think about oxidation numbers, but can be interesting to look at. So in peptide bond formation, you have this carbon in an amino acid, the carboxyl group of an amino acid, that is joined to this nitrogen. That's the peptide bond. So it will look like this. And this carbon initially in the free amino acid is carboxyl, so it has +3. In this case, you have exchanged one oxygen for a nitrogen. So this oxygen has been exchanged for this nitrogen. And as we said, oxygen and nitrogen are almost the same. So if you put the formal oxidation number of this, it's going to be +3 still. So you can't really say in which direction this is going to go. But it's a borderline case. As you remember, we said that the nitrogen atom is a bit less electronegative than the oxygen atom. So you might think that it's borderline, but you might think that this is unfavorable because the oxygen sort of is a better choice here than the nitrogen here for the carbon atom. And that's indeed the case. Peptide bonds require energy to form. So you need to have a GTP in this case to drive this reaction. So those are some examples. We can also sort of think of this as a perspective of metabolism. So redox gives us a way of looking at and organizing metabolism on kind of an axis from a reduced state of metabolites to an oxidized state. And actually, I think many of the metabolic pathway charts that you see are kind of drawn with this in mind. You typically see glycolysis, for example, being shown as going from sugars on the top down towards pyruvate and lactate at the bottom, and that's the direction of oxidation. So it's kind of an intuitive way of drawing these maps that you are kind of falling down in energy from the top to the bottom, and CO2 is sort of at the bottom. So this is a chart that I drew and contains some of the main sort of components of metabolism, the main classes. And you can find sugars and amino acids somewhere in the middle. You will find the more oxidized things like pyruvate, acetyl-CoA, central metabolite, which contains ketone groups in the middle. Further down, you have carboxylates and, again, CO2 at the bottom. So that's one useful perspective sort of to keep in mind when you study your pathways. Where are they on this type of chart? So just one final point is that, as we said in the beginning, these electrons that are transferred between all sorts of atoms in redox metabolism, they are of course carried by something. We'll look at this in a bit more detail later, but they are carried by specialized cofactors, energy electron carriers like NADH, NADP, NADPH, FAD, FADH. And one interesting thing with this, which has been observed consistently, is that oxidation and energy metabolism on the left here uses a different set of electron carriers than reductive metabolism, biosynthesis. So typically we see that NAD and FAD, to some extent, are the main carriers in energy metabolism and oxidation of things. So when a cell wants to take a substrate and oxidize it and extract energy, these are the electron carriers used. When you're going in the opposite direction and a cell wants to take a substrate that is a bit oxidized and deliver electrons to it to do reduction and to do biosynthesis to reach a reduced product, maybe synthesized fat or synthesized membranes or synthesized nucleotides, then it is using a different electron carrier. It is using NADP. So NADPH, the reduced form, is converted to NADP, and this power is used for reduction. And this is an important point, I think, because this compartmentalizes redox metabolism. You have two compartments. You have the oxidative and you have the reductive compartment. So two sort of separate processes are occurring at the same time. And this has to be probably like this because all cells have to do both oxidation and reduction at the same time. All cells need to oxidize something in order to extract energy. At least all animal cells. If you're a plant, you have other options. But all human cells that we consider have to do this. And at the same time, they usually want to synthesize something. So we always have oxidation and reduction occurring at the same time. And these different carriers help to separate the processes so that the cell can control these things independently. So that's an important theme in redox metabolism.